So the project we are involving in since last 2 years is using wild relatives of rice to breed new varieties which are more resistant to unpredictable water availability. And this is a part of the initiative, one of 11 projects, an initiative called Sustainable Crop Production Research for International Development, which was aimed at improving food crop productions in developing countries in South Asia and Africa. So why, this is not a map of UK, this is a map of India. So why we need to have drought-resistant rice in India? So if you look at the map on the left, which shows you the percentage of the rice grown in the paddy fields, they dominate in the south of the country and north of the country. But in the middle, you have a huge belt that is growing upland rice, which means the rain-fed conditions, which means that rice depends only on the rainfall. Productivity of that rice depends on the rainfall. And if you compare that map with the middle one, which shows the productivity of these regions, you see showing the paddy fields are giving a high yields on the south and the north. But the regions of the upland rice growing are very low in productivity. If you compare that to the last map on the right, the regions in the middle belt are prone to drought, and it's actually around 20% of the total grow area of rice in India is prone to the drought. This message is actually reflected in that slide, which is 10 main causes of losses of yields in eastern India, which is represented as millions of tons lost every year. So you see the major factor in the yield losses is drought during the flowering, during anthesis when they're setting the seeds, but also quite vulnerable stage is seedling stage. For example, if the drought happens in between the seedling and flowering stage, it doesn't have that much effect on the yields. To point out also submergence, which is flooding, is also another big cause of the losses of the yields, together with some other bacteria or other biotic stress, other diseases basically. So why is rice so susceptible for drought? Actually, domestication of rice happened around 8 to 13,000 years ago, and we had the huge variation in the wild, huge genetic variation in the wild species, which then there were through the intensive breeding went through what we call domestication bottlenecks, which actually causes a loss of the alleles, and those alleles conferred the resistance to all these diseases and drought, for example. At the end of the process, with the modern mega yielding varieties we have at the moment, we started with something which looked like a grass, and now we have Chinese hybrid rice, which is reaching 14 tons of grain per hectare. It's amazing yields. But on the way, we lost all this diversity here, and that's why you have a problem of drought resistance, drought sensitivity in rice. And how can we? There is a clever way of introducing this white alleles back to these commercial varieties called chromosome segment substitution lines, where you basically backcross your favorite variety, high-yielding variety, you cross it to the wild relative, and then in the series of back crosses and selfing, you end up with the lines which have just a small proportion, just a small fragment of the genome, which is represented here in the color, is coming from the wild rice, from the wild ancestor, and the rest in all the 12 chromosomes is the background of your high-yielding variety. And then if you align these lines, the collection covers basically all the genome, entire genome of the wild ancestor in the very small pieces. So our project uses two wild ancestors, Oryza, actually three. Oryza meridionalis is the wild rice from Australia. It's very sensitive, it's very tolerant for drought, high aluminum content, and some bacteria disease. And Oryza rufipogon, which is direct ancestor of rice from Asia, also shown to be resistant to diseases and drought. And the background we use is the commercial drought-tolerant upland variety of Coringa, developed in Brazil. So that's the important message from the previous talk, because if we would lose these wild ancestors, if you would lose these species, then we wouldn't be able to create these collections, which are very valuable collections for breeding. That's the material we use for our project, and this is the is international the initiative really, because we have Cornell University in US which developed the germplasm, and we have the field trials happening in India in a place called Cuttack in Central Rice Research Institute, which are very experienced in rice breeding, and they have fully irrigated and drought condition sites, which I'm going to talk later about. And we in York, we actually try to understand why these lines are resistant at the molecular level. So we do phenotyping using hydroponics and soil system, which I'm going to talk about later. We measure also phytohormones and metabolites. But also the Cornell University is doing three-dimensional root architecture screen, and we also do a little bit of socioeconomic studies and climate modeling, which I'm also going to talk about later. So that's how it looks like in India, in Cuttack. We have our germplasm grown in the full irrigated fields and also in what they call rain-out shelters, where basically you can impose the water stress, and they do it over the flowering time, just start before flowering, and while they're setting seeds. And so they score multiple traits, a lot of different traits during this process of field trialing, but the ultimate goal is to get the variety with the little or no reduction in grain yield under stress. So if the same thing is grown in fully irrigated condition and under stress, it shouldn't lose the grain. It should produce more or less similar amount of the grain, and that's how it's defined to be drought tolerant. So we have in over 2 years data collected from the 3 sites. They have also upland farm with the real field conditions there, and we have the data from there are 2 growing seasons, so we have 2 wet season and 2 dry season data available. What we do in the lab, we grow plants hydroponically, and we use PEG to induce osmotic stress at the seedling stage. We try to operate at quite low concentration of PEG to be similar in the range of the osmotic pressure to the field condition. And we're looking at, and PEG is inducing, it's resulting in typical responses of osmotic stress, like in decreased shoot biomass and increase the roots. So basically shoot stop growing, roots starts growing after imposing PEG. Leaves start to roll, so the leaf width decreases, and stomata closes to prevent transpiration, and then ABA concentration increases. What we also look at is the metabolites. We do Metabolomics, GCMS, where we can measure about 300 metabolites. This is just a selection of few of them. And we're looking, so the first is PEG. So you see the PEG induces the responses in increase in major osmolytes like sugars, glucose, fructose, sugar alcohols, or organic and amino acids. So we're looking at the lines like for example, line 48 that is actually mobilizing the osmolytes very quickly and to the higher extent than the recurrent parent, and therefore it's maybe better equipped to survive the water stress at the seedling stage. We have the other activities of the project. I told you about 3-dimensional root architecture screen. The collaborators in Cornell growing these lines in kind of gel-on gum filled cylinders, and you can also impose the water stress, osmotic stress by PEG. You can monitor the root growth 3-dimensionally over 2 weeks. And yeah, it's a good system, but when I heard today about Malcolm's system in Birmingham and the micro CT facility there, it will be quite good to use something like that for root architecture screen as well. And what I want to say, the background is not that important. This recurrent parent is not that important because what the people in Syria will do in collaboration with Cornell, they're going to introduce these white alleles. You can do it with following them by molecular markers, by SNPs, and you can cross and then do the marker-assisted backcrossing to the own variety, which are the favorite 5 varieties. But we're also using that as a recurrent parent also 2 lines, IR64 sub-1 and Swarna sub-1. So sub-1 causes the resistance to submergence. So then you have a field here where it's shown what happens to the yields after the plants being flooded, and then water is gone. It's basically there is no grain anymore. Whereas sub-1 can survive submerged in water for over 2 weeks. I bet that farmers in Somerset would appreciate to have a wheat variety with sub-1. But it's also introduced from IR64 to the Indian variety Swarna. I don't think that the selection of the farmers was random in that picture. So the guy on the right is growing Swarna sub-1, which basically yields very well after the submergence. So in this way, hopefully we have variety which is drought resistant and also submergence tolerant at the same time, because the drought and flooding can happen in the same field in the same season, more and more likely. What we also do was a little bit of climate modeling, so we see how much the precipitation will change over time, how much the monsoon precipitation is changing. Break dates in monsoon period are quite important for farming. And finally, we work with Siri works with farmers through the discussion groups, questionnaires, just to see what the farmers want, because there is no point of developing the new variety of rice where your farmers at the end say, we don't want to grow it because we don't like it. So we need to know what they really are interested in, what are the characteristics they really like in these new varieties. And that's all the socioeconomic studies we do in the project. So a lot of thanks to all the big team from York. Ian Graham is PI of that project. I work a lot with Tony Larson on metabolite profiling. Lin Chen is a postdoc I'm working with together quite a lot. And the people from Siri, we're going to host them next week in York. And the team from Cornell also coming to York for the meeting. That was a very short overview. If you want to have more information, please visit the website of our project. And thank you for your attention.